button up. Everybody to button up. Can button up. Okay. All right. A very good afternoon, everyone, to the School of Life Science and Chemical Technology. Okay. In LSCT, in short, we offer a few diploma courses, and today we are having a mini workshop in the Biomedical Science diploma. All right. So we have more knowledge about this particular diploma. Right. So a lot of students who sign up for BMS, they aspire to save lives or have solutions for some of the common healthcare issues. For example, they are trying to understand what actually in the grandparents, the gene in the grandparents, how come they can cause, let's say, cancers in the grandson? How come every year we keep seeing new diseases that come out? Like 2 years ago, we have COVID-19 that actually bring the whole economy to a stop. Okay, all the human activity to a stop. All right. So, and then our students will try to think of new or innovative idea how we can actually provide solutions for all sorts of issues like this. Okay, so in this case, biomedical science diploma, in this case, we train our students in the following fields. For example, research and development. Can we come out with a new treatment for a disease? For example, something simple as flu, hundreds of years from now, we still cannot totally get rid of them. Is there a new way of how we can go through R&D to develop new weapons against them? All right, and can we mass manufacture certain drugs like vaccines? Before COVID, as you know, Singapore have 0 vaccine manufacturing plant. But after COVID, now we have 5. And they employ hundreds of employees that have specialized skills or knowledge to mass produce vaccines. So as other type of biologics or biomolecules, such as hormones, antibodies, stem cells. All these are new generation of drugs that help us to cure the diseases. Okay, and with the knowledge of life science, it will allow you to also work in the food manufacturing. Okay, like very traditionally, we have kimchi, we have wine, beer, and all these different fermented products, or even chocolates. They are all produced by the help of microorganisms. All right, our students will also have a chance to learn how to assess a food. Like for example, a bread that you keep from last week till now, are they still safe for consumption? So there are a few tests you can do to actually extract the bacteria there and see whether they are harmful or anything growing inside. Okay, so that's on food manufacturing, and then we also have clinical diagnostics. Patients that say it comes to you, the doctors have fevers, cough, sore throat, and runny nose. All right, so do you think that the doctors will know what infects the patients? The answer is no, because more than 200 types of microorganisms can cause exactly the same symptoms. So you just collect some of the samples from the patient, and with that, we'll send to our graduates working in the clinical diagnostic lab. They'll run through a serial diagnosis, and they will find out what actually have infected the patients so that the doctor can prescribe the right medication. All right, so these are the many different areas our students are trained in so that they have a very versatile career, all right, later on after they graduate. All right, so this is how the course are structured over the 3 years. You can see that from year 1, all students will start with the foundation knowledge or modules. For example, how the body functions, and then these are on the cells and our genetic materials, DNA, RNA. Okay, and then they will learn about microorganisms, virus, bacteria, fungi, for example. All right, so even those without any biology background, not to worry, you can step in to our year 1 confidently, because we'll build out from foundation, so that everyone will have arrive at the same page before we move into year 2. All right, so for year 2, it's mainly more on deepening of the module and on focus on the applied aspect of this module. For example, you learn chemistry here is applied chemistry. You learn microbiology here is applied microbiology. Just following on the food example, like microbiology here, you learn about how to make media to let the cells grow. How do you actually test the bacteria, find out their identity? So applied means that you actually use the microorganism, that the skills you learn on microorganisms, and then you use them to either detect them in the food, or you may actually carry out fermentation to produce a food with different flavors. Okay, so in this case, that's on food technology, food process safety part that you'll be learning if you choose the food track. Okay, and also just what we mentioned, the new generation of medicine is no longer just relying on pure chemistry. All right, in the past, if you have a headache, you actually pop a Panadol. A Panadol is made of paracetamol, which is chemically synthesized. But new generation of medicines includes hormone, antibodies, stem cells, which in this case, we need cells to grow. All right. For example, a patient who has diabetes. All right. And in this case, for diabetes, how do we treat the patient with diabetes? We give them something like insulin. Excellent. All right. So with the insulin, in this case, how do you get insulin? As you know, it's a hormone. Do you kill another human being to save another human being? No. All right. So what you do is that you take the human insulin gene, but you put it into a bacteria. And then you grow this bacteria in bioreactors in huge quantity. All right. And the bacteria will now become the mini factory. They'll release the hormone into the culture media. And then you purify that hormones. And that is actually the medicine to treat diabetes. All right. And similarly, there are many issues with health that you can use antibodies, even use, for example, antibodies therapy for cancer treatment. And the new generation, we have CAR T cells. We actually take out cells from human body, genetically modify it, and then from the cancer patient's body, genetically modify the white blood cells, and then put it back into the same patient. Because our immune cells are not naturally not that effective in fighting certain cancers. But now, if we modify the cancer cells, the white blood cells, and put it back into the body, they are like our soldiers with enhanced capability. They'll search through our body to find the cancer cells and eliminate them. So all these are new generation of treatment, which the students will learn under biomanufacturing. Okay, so the different ways that we produce new therapy and what are the different requirements and the technology. Okay, that's under biomanufacturing. And then in the third year, we move into the real world situation. All right, so in this case, the students will continue to deepen their knowledge in applied biomedical science track. They'll learn about genomics, proteomics, translational medicine, clinical trials, or many such modules to help them deepen further. And at the same time, we help them to connect the dots. Because you can see here, there are 20, 30 modules here. But how do you put the knowledge together to develop a solution? That's what a final year project is doing. We call it capstone project. So the students will actually carry out a research project for half a year, either in Ngee Ann Polytechnic or in our partnering company. All right, and with this, in this case, they will apply the knowledge that they learn in year 1 and 2 to solve problems or create some products. All right, and then the next semester, they will go on for internship, 6 months. They can choose areas such as, for example, drug manufacturing. They can go into, let's say, for example, if they are more interested in patient care, all right, diagnostics, they can also work in healthcare, all right, institutes. If they like R&D, in this case, they can go into A-star or universities or research lab. So these are the different, many different companies they can go in, all right, for their internship. Of course, if some students who are particularly interested in patients' care, all right, or for example, clinical trials and also diagnostics. In this case, they can go in to take this special program. It's called Integrated Clinical Training Program, all right? And that's for 1 year in Singapore General Hospital. So over that 1 year, in this case, the students can choose an area of their interest of, for example, these 5 areas. And they learn from the staff there, SGH staff, and then immediately they apply it on the job. Okay, so just for example, clinical trials, if someone come out with a new medicine, how do you test whether this new medicine is really effective? How do you, then the existing medicine, all right, and whether there's any side effects, so on and so forth. So you can actually learn the procedures through here. Or if a patient sends in their samples, you can carry out different type of diagnostic tools and techniques to help you identify what was the patient's problem, underlying health problem. All right, and in this case, you can help that with the diagnostics. Okay, for the rest, you can just Google and see what that means. All All right, but these are the different areas you can learn from the 1 year training in Singapore General Hospital. Okay, at the moment, is there any question? Okay, all right, if you don't have, then these are the different type of careers. Upon graduation, because diploma will train you to be job ready. All right, because let's say one module, let's say microbiology, you take 2 hours of lecture online, and then 1 hour's tutorial per week. And that tutorial is to help you make sure that, help us make sure that everyone in the class is in the same page. They understand the online lectures. All right, and then after that, they will do 3 hours per week of hands-on. In this case, they will learn about how to culture cells. They'll learn about how to identify the bacteria, test for antibiotic sensitivity, so on and so forth. All right, so with all these skills, our students can actually have a good dynamic careers in all these different roles. They will be very familiar with data analysis. They can work in food company or industry or sector. They can go into healthcare setting and help with clinical trials or actually carry out diagnostics. They can also work in quality assurance system, quality control analyst. All right. They can also go into companies, Pfizer, GSK, Merck, that manufacture drugs, vaccines, hormones, and they can go there and make medicine. Okay. So many different areas they can go in. All right. Because our curriculum make them very versatile with the skills that they have. Right. Of course, not all students want to immediately start with their career. Some of them would like to further their study to get a degree. All right, and then no issue, you can see that more than 80% of graduates will go into university and complete their degree course. About 30 to 40% in the local U, the other 40% in overseas universities. All right, so why many also choose overseas universities? Because they can get advanced standing. All right, let's say, for example, you take a biomedical science course, you completed your microbiology module with A1, flying colors. All right, and then when you go overseas, let's say University of Queensland, you also take biomedical science or life science, they also run microbiology module. Then they will say that since you're already trained in Ngee Ann Polytechnic, you don't have to do the same module there. That's why you can get up to 2 years of exemption from these overseas universities. Okay. Of course, there's many of you, our graduates can also go into local universities, and usually they get half a year to 1 year of exemptions. All right. So we also have graduates who go directly into medical schools upon graduation, and so far we have 36 of them that go from poly directly into medical schools. All right. So just now we mentioned many different aspects of where our graduates can work in. And now I want to throw in a case study on this thing. All right, make a guess. What is this thing that I portray here? Anyone? You want to just sit inside here? What is this entity here that I'm showing here? Make a guess. Something close to us. Which one? Coronavirus, the COVID-19 virus, okay? All right, so in this case, when COVID was first break out, if I just show you this picture, can you tell me whether this is a new virus or is something that we already have, like it's SARS, or it's a flu? Are you able to tell by just looking on a picture like this? The answer is, of course, cannot. How do you tell whether this is a new pathogen, a new virus, or is an old one? Old one, we feel safe because we know how to deal with them. But if it's something new, we need to have a new strategies. All right, so the first thing is to find out whether this guy is a new one. How do you find out whether it's a new one or not? We look at this thing, the genetic part of it. All right, so with the genetic part, in this case, we carry out gene sequencing. That tells us that, oh no, this guy is actually a new virus called SARS-CoV-2. It's not SARS that we have handled more than 10 years ago. So we need a total different two kits to deal with them. All right. So so that's how biotechnology come in to help. It helps us identify that, whether this is a new virus. And now a patient come to you and say that I've runny nose, I've sore throat fever. Am I infected with flu or am I infected with COVID-19? Okay. So at the very beginning of COVID-19, what was the method we used to do diagnosis? Anyone still remember? Start with P. PCR, okay? So for PCR, in this case, it's also a genetic method. We actually put in a probe right to the back, all right, near the throat there, to do some scraping, so that you can get a bit of the virus genetic materials out. And with that, you run PCR, and in this case, you can confirm that, oh, this patient is flu or it is infected by COVID-19 virus. Okay, so after that, now no longer we do this. Why? What's the new methods we use to do diagnosis at home? Something you start with A? ART, excellent. ART means that we are trying to detect not the genetic material, but these viruses also have very unique proteins on the surface. Okay, so we carry out antigen detection, and we just do this, and you put a probe. Now you don't need to extend it all the way to the back of your throat. You just do it in the nasal part, and immediately within minutes you can get the result. Like this one, you need to wait 1 to 2 days. All right, earlier. Now you get within minutes. Okay, and then next, how do we prevent if we haven't catch the virus? How do you protect people from catching the virus? What are the tools that we have to protect people from catching the virus? Something starts with V. Vaccine, excellent. All right. So with biomedical research R&D, in this case, people come out with vaccination. Vaccines is by actually using the RNA to inject into the patients, so patients develop immunity against the virus. So even if they get contracted the disease later, infected by the virus later, the symptoms will be much weaker or less. Okay. So, but how about if someone too late, they did not get vaccine, they are now very sick, dying? Do we have a solution for them? We say that, or we just say that you just drink more water. Is there a solution for them? Answer is yes. For biomedical science, scientists also develop medications such as Paxlovid. In this case, it's only prescribed for patients who have very serious COVID symptoms. All right, just to share with you that you can see here, BMS plays central role in fighting one pandemic like this. All right, there are different solutions targeting from the virus from different angles. All right, and then for today, we are going to focus on just one, the molecular testing part of this virus using this method called PCR. Now, one part of it, PCR, what is the full name? Anyone know? It stands for what? What does it stand for? We keep saying PCR, but sometimes we may not be actually forgotten that it actually have a full long name. Anyone want to give it a try? Yes. Wow, is there somewhere you can see? Oh, okay, sorry, I forgot to block this one. Yes, all right, so it's actually polymerase chain reaction. So I know that for some of you who are interested to apply for BMS course through EAE, that is something that we may ask you, all right? Because everyone will say that they are interested in life science, in biomedical science. But how do you show that you're really interested? It means that for current issue like this, advanced development in biomedical science, biotech, you actually follow up closely. You are knowledgeable about things like this, polymerase chain reaction. All right, you are knowledgeable about, the vaccine is actually using RNA and how your immune system works to fight back against the virus. Okay, so those are the tips for those who are actually planning to apply biomedical science through EAE. All right, beef up on your knowledge in terms of biomedical science. Right, so yes, for PCR, in this case, we are looking for this unique gene called N gene inside there. Because this N gene of COVID virus is different from that of any other virus. It's something only unique to COVID-19 virus. Okay, so we are going to use this method, technology, to make it into a million copy. What do I mean? For example, if you do a swab on the patients at the back of the throat, and then you take out the cotton swab, do you think that we take out a lot of DNA from the patient? No, it's very, very tiny about. Even if you stare into that cotton bud, you can't see anything. All right, that's how minute the sample is. So the first thing we need to do is to put the genetic material of whatever you get from the patient's throat through a so-called photocopy machine. And that photocopy machine copies genetic materials into millions copies within a short period of 1 to 2 hours. All right, so this photocopy machine will make billions of copies so that you make the DNA visible. Okay, so when we talk about genetic sequence, what comes to our mind? genetic sequence, about DNA, what comes to your mind? Just now I talked about, oh, the genetic sequence of COVID is different from bacteria. It's different from flu, different from SARS. What type of sequence are we talking about? Anyone know about what DNA is made of? How many chemicals make up DNA? Any chemicals? Clue is how many types? 2 types, 4 types, 8 types. May I guess? How many? 4 types. All right, the answer is yes, 4 types. And these 4 chemicals is just like this. All right, this is a full sequence of the SARS-CoV-2, COVID virus genetic sequence. There are close to 30,000 base pair. And this is not even 1/3 of it. It's just a small portion. But this genetic arrangement, you can see that there are just 4 sequences, A, G, T, and C. These 4 chemicals. But in different sequence, they actually join up together that give the cells, the virus, the instruction to make more COVID-19 virus. And that's what we call the blueprint of life. All right, humans have our own unique sequence as well, and our cells get instruction from there to make human cells. Virus get instruction from their unique sequence to make more virus. Bacteria get unique sequence to make their own. So by detecting the different types of sequence like this, we can identify the virus. Okay, so that is the principle of PCR. Make a guess. For example, if I have a piece of paper, imagine this is a piece of paper. If I have a piece of paper like this, if I want to photocopy this paper, what do I need? For the photocopier, what do you have in the photocopier? You need at least 2 things. The ink plus paper. Okay, if you want to make photocopy of that, you need ink and paper. All right, same thing. If you want to copy genetic materials, a piece of DNA from the virus, you also need first original copy, which come from the patient. You do a throat swab, the patient sample is original copy. Then you have the ingredient, which is the ink. All right, the 4 chemical, C, G, A, and T. All right, of course, you need a photocopier machine. And in this case, these tools that copy the genetic materials is this enzyme. All right. So, and then also we have something unique called primers that will help to look for the unique sequence in the virus and binds to it. Okay. So after that, we put it into this machine, and this machine will help us copy the genetic materials by going through heating, let the primers here to bind to it, and then cooling, and then let this green copy machines to copy the genetic materials. You can see 1 into 2 copy. After a few rounds, it becomes billions of copies. All right. So this is something we call polymerase chain reaction. Okay. Any questions so far? Right. So this is what we are going to do today. And of course, not on the PCR part, because that may take a short while more, like hours. So today we are going to focus on one part. We already run the PCR for you. Let's say if I'm going to show you 2 tubes like this, are you able to tell me which tube contains the virus and which virus genetic materials and which one don't have? Are you able to tell? Cannot. Even if I pass to you, you stare at it for 1 hour, you still cannot tell because they are both still 2 droplets of liquid. You can't see them at all. Okay, so we need something to help us see which one actually have their genetic materials copy, which one don't have. Okay, so that is what we are going to explore today. And later on, this is what we call the gel. And then we have it here. Okay, I'm going to put on gloves and to open up one gel. Okay, so basically gel is made of a specialized, it's made of the same agar-agar you take. The agar-agar we use to make kueh, all right, or different type of what's it called, jelly during festive seasons. All right, it's the same material, and then it's semi-solid. And then in this case, we load the DNA samples into something like holes on the top. Okay, so this gel looks like this. I'm going to tear it open. Okay, you can see that it's a small little block with little holes on the top. All right, and then it is semi-solid. It has a thickness. So you can see the holes like this. So later on, what you are going to do is that you are going to take a drop of the patient's sample. These are what we call micropipettes. We already preset it for you. Later, I will give you a chance to explore how to use it. So for these micropipettes, you are going to put a tip to it, okay, and then open up this, take out the sample from here, all right, and then use this to load into this sample, okay. After that, we'll put this gel into the machine here, and the machine will actually apply an electric field, and the genetic materials will be separated based on the sizes, okay. It's just like a race. The shorter piece of DNA will run faster. The longer piece of DNA will run slower, okay. So in the end, you see bands like this, okay. So if a person with a virus PCR products show up, the bands, DNA bands, we know that this patient is infected. If no bands, we know that like this, if no bands, in this case, it's not infected. That's how we do the diagnosis, all right? So what you learn is actually the clinical diagnostic part using the molecular tools that we have. Okay, so far, any question? Okay, don't have, then next part, we'll go into how to use the pipettes. This thing is what we call micropipettes. Okay, for micropipettes handle volumes that is 1 over 1,000 of milliliter. Imagine 1 milliliter, but micropipette allow us to handle volume that is 1 over 1,000 of per 1 milliliter. Okay, we call that microliter. Okay, so today we are going to use 0.02 milliliter of the sample from the patient. All right, and then we are going to load it into this well. So first on how to use this pipette. This pipette, in this case, can each of you hold a pipette? Can I pass to you? Just a while. I'm going to pass the pipette to you. Oh, you just. Okay. All right. So you have a pipette, and then it's already been preset for you already. All you need to do is to take out the sample, but there's also a way to take out a sample. First, can you press like this? 1, 2. 1, 2. Okay, you find that there's 2 levels of resistance. The first level is actually easier to press down. Okay, then the second level is harder to press down. We call this the first stage, second stage. Okay, so this is how you take out the sample. Right, you cannot just use the pipette as it is naked. You need to take a box like this. We call that pipette tips. And then gently plug in the pipette. Give it a firm push so that this pipette actually have an airtight seal at the tip. Okay, try not to do something like this, because that will, if you do it many times, then the pipette will misalign inside, because it's very delicate and accurate. It's a very precise equipment. All right, so just give it a gentle press. Make sure that this thing is an airtight seal, and then you have a tip in front. Okay. Next, I'm going to randomly give you some sample. Some contains patients' DNA that have the virus, some without. Patients, all right, so you don't know which one it is until we run the gel and you analyze from the data. Okay, so this is what you are going to do. You press until the first stop. Go in, hold on to it, and then you go in. Make sure that you see the tip come into contact with the patient sample, and then you take out, and then you slowly release it. Okay, so now the patient's samples or the PCR product has been taken in to the tip. Next, you are going to this gel, and then you are going to load in the gel. All right, when I'm going to load in the gel, I'm going to just show you how we load in. Okay, so just I don't want to waste the gel because you are supposed to look in this one. All right, so I'm just loading it on my hands here for example. Okay, so how to load the gel? First, you find the well, okay, like this. Go directly into the well, okay, and then you press to the first stop, not just the first stop, but second stop. In this case, you push out every last single bit of the sample in the tip. Okay, so this thing will now go into the well. All right, confident to do that? All right, so in this case, that is how you load the gel. Okay, after you load the gel, then in this case, we'll take the gel, put into this electric field, and let it run. and then you will see whether your samples actually is from the patient that is infected or from a healthy patient. All right, okay. All right, so let me see. So you have learned about the use of micropipettes. Anyone have question or still not too sure on how to use it? Can, All right. Then now we will get started. So we'll all move over to that table. This is where we'll load the gel. Can you guys come to my side? All this can remove. Okay. Okay, I'm going to give out. All right, okay. So we have some samples here. Okay, so you just choose any sample. And then later on, we are going to, can you all come over to this side? So that they can still see. Okay, yeah, you can stay here. Okay, so choose your sample because now we don't know which one contains from the sample with patients infected and some without. Okay, you choose S1. What's yours? Okay, S6. How about? Oh, have you put on gloves? Oh, sorry. You need to put on gloves first. All right. Yes. The gloves you can take from there. Okay, those of you who are already done your glove, you can come over. Okay, gloves is a very important tools that we use to protect ourselves. All right, so that we don't get infected during any of the experiment. Okay, so now I just randomly distribute the samples. All right. You can just stay here. All right. All right. Okay, now can you take a hard look at your sample? Can you tell me whether your sample contains the virus genetic material or not? But I already copied millions of times already. There are millions of copies of the virus genetic material. Still cannot see? Answer is no. Okay, that's why we need this to help us see which one carries the virus genetic material. So you just open it up like this, and then you can all take your own pipettes, give it a firm push. Okay, and then go inside here. Make sure that you go all the way to the bottom and slowly release the plunger. All right, so you can see that all the liquid are already in the tip. Next thing, you are going to just do like this, stabilize your elbow, and then you are going to load into one of the wells. Okay, I'm going to remove. Just a while. Okay, so this is the first one. For example, I just load inside like that. Okay, and then after that, this one can be thrown off. All right, by pressing here. Okay, all right. So now you can see that your sample. Remember, my sample in this case, I load into well M. Okay, so what's the number of the well you loaded in? You need to remember. So later you'll see. Okay, can you give it a try? Yeah, just press down. Press firmly. Okay, just one. I want to make sure that. Oh, you use this one. This, yeah. Did every drop goes in? Okay. All right. For those of you who are waiting, you can get your own pipette and start pipetting it from your own sample. I think just now this part was not, it was loose. So you have to make sure that it's airtight. Then you try again. Okay, press to the first dot and then go into the tube and then slowly release. Great. All right, now you can load the sample. You are load into number 4. Okay. Wow, excellent, so fast. Okay, you can eject. Yeah, okay. So got 4, yours is 2. Okay. 4 and 2 taken up. You do 3. Okay. Okay. Don't release, hold and then lift up. Okay, excellent. All right. We let them have. Okay. You can see that. Number 1 is still available. You can load into 1. Press all the way. Okay, excellent. Number 5 is still empty. You want to take number 5? Oh, I think your samples, some of them are still inside. Yeah, you can see there's some inside. So you can just put them back first. Press all the way down. Okay, now you hold like that. So now I'm going to go all the way to the bottom and take out. Release. Okay. So you can go number 5. Anyone else haven't loaded or all loaded? You mean no? Oh, empty. Okay. All right. So, is there any more Oh, can we have more sample? I think one of them haven't loaded yet. Okay. You want to? Okay. I'm going to fill up all the others unused wells with just water. Just now you loaded into 5, huh? Okay. You load 6. I'm going to fill up the rest. This is just water. You can put into 6. Excellent. Okay. All right, can throw it here. So now I'm going to throw in all the samples. And okay, so now we have this machine. This machine is going to apply an electric field so that our genetic materials will start moving from here to the bottom, all right, to allow us to see which one will give you a band, all right? So I'm going to open up this machine first. Okay, and then I'm going to load the gel. Okay. Next, I'm going to connect it to a PowerPoint and turn on. Is this? Oops, sorry. Is it running? Okay. Okay. Oh, okay. Now it's live. Okay, I'm going to go the other side. Okay, I'm going to set up run. Let me see. Okay, so I have 11 wells. This is a 1% gel. Okay, I'm going to try 10 minutes. But before I start, I'm going to put a viewer here. Okay, so basically this will allow you to do real-time viewing of the gel. You can come over here and take a look. Okay, setup run. Next 10%, start running. Okay, so now I have pressed start running. The electric field has been applied. So in this case, I can press view gel. Okay, so now nothing here because all the samples are in the well. All right, so we are going to see. Do you start to see something? They start to get attracted into the gel matrix. Okay, so we are going to just wait 5 minutes, and along the way, you'll see that which are the patients that have been infected by the virus or COVID positive. At the moment, do you have any question about this particular diploma? Is the course difficult? All courses are difficult. All right. So no, what happened is that they have certain knowledge. I'm sure that for any discipline, if you want to really go into innovation, you want to really help patients or develop something useful, there are some basics that you have to understand and grasp. All right. So those are the parts that you make sure that you actually have a good grasp of the foundation, and it's not really that difficult to pass the module because we don't have one killer exam in the end. All right, we actually split the course into many components, like for example, assignment, workshops, worksheets, projects, presentations. So as long as you actually complete the assignment in time and submit, in this case, you will be able to score them well. All right, you will not be able to fail it. We don't have one killer component at the end of the semester. Okay, so now you start to see something in the over here already. Okay, just now which sample have you loaded? Oh no, sorry, which well? Well number 1. Yeah. So can you tell me well number 1, the patient, is it infected? The patient that's coded called S1. Is the patient infected or not?
I think yes.
You think yes? What gives you the conclusion? The answer is correct. All right, but how do you derive that?
Because it's more like visible.
Yes, it shows you a band. A band means that it's a positive test. All right, it means that the virus genetic material was actually in the patient's throat. How about yours? Your sample? You look in well number 2. Okay, so for number 2, was your patient infected? No, because you don't see any band there. So this is number 2. I can actually. Okay, all right. So, yeah. So how about yours? 5. How's the patient? Healthy or not healthy? No. Okay. No, because you see a band. All right. So how about yours? Infected. Which well is that? 6. Okay. All right. And you can see that because of the different viral load, so some of them have a thicker band, like patient number 6 and patient number 1. All right, have a thicker viral load because indicating more genetic materials from the virus are there.
Number 4, it has a pink color.
So for what do you guess for number 4? Infected. Okay, but at what stage?
The late stage because the viral load is very low.
Viral load is low. It's either at the very beginning when the virus start to multiply, or at a very late stage when your body already killed off majority of viruses.
So it's either beginning or end.
Yes, but no matter what is that, in this case, the person is infected. So that's how we draw conclusion. And this is what we call clinical diagnostics. All right. Okay. So now the result is quite clear. So we can see that patient, those who look into 2, sorry, is that 2? 2 and 3 are actually free from the virus. Okay. All right. So that will round out our experiment today. I hope you bring home something useful. Okay. Thank you. Bye-bye.